A taste receptor is a type of receptor which facilitates the sensation of taste. When food or other substances enter the mouth, molecules interact with saliva and are bound to taste receptors in the oral cavity and other locations. Molecules which give a sensation of taste are considered sapid. Taste receptors are divided into two families. Type 1, sweet, first characterized in 2001, TAS1R2 TAS1R3. Type 2, bitter, first characterized in 2000, TAS2R1 TAS2R50, and TAS2R60. Combinations of these receptors in dimers or other complexes contributes to different perceptions of taste. Visual, olfactive, subjective, the perception of tastes, trigeminal, hot, cool, mechanical, all contribute to the perception of taste. Of these, transient receptor potential cation channel subfamily V member 1, TRPV1, vanilloid receptors are responsible for the perception of heat from some molecules such as capsaicin, and a CMR1 receptor is responsible for the perception of cold from molecules such as menthol, eucalyptol, and isolin. Tissue distribution. The gustatory system consists of taste receptor cells in taste buds. Taste buds, in turn, are contained in structures called papillae. There are three types of papillae involved in taste: fungiform papillae, foliate papillae, and circumvallate papillae. The fourth type, filiform papillae, do not contain taste buds. Beyond the papillae, taste receptors are also in the palate and early parts of the digestive system like the larynx and upper esophagus. There are three cranial nerves that innervate the tongue: the vagus nerve, glossopharyngeal nerve, and the facial nerve. The glossopharyngeal nerve and the chorda tympani branch of the facial nerve innervate the TAS1R and TAS2R taste receptors. In 2010, researchers found bitter receptors in lung tissue, which cause airways to relax when a bitter substance is encountered. They believe this mechanism is evolutionarily adaptive because it helps clear lung infections, but could also be exploited to treat asthma and chronic obstructive pulmonary disease. Function. Taste helps to identify toxins and maintain nutrition. 5 basic tastes are recognized today: salty, sweet, bitter, sour, and umami. Salty and sour taste sensations are both detected through ion channels. Sweet, bitter, and umami tastes, however, are detected by way of G-protein-coupled taste receptors. In addition, some agents can function as taste modifiers, as myraculin or curcumin for sweet or sterabin to mask bitter. Mechanism of action. The standard bitter, sweet, or umami taste receptor is a G-protein-coupled receptor with 7 transmembrane domains. Ligand binding at the taste receptors activate second messenger cascades to depolarize the taste cell. Gustducin is the most common taste G-alpha subunit, having a major role in TAS2R bitter taste reception. Gustducin is a homologue for transducin, a G protein involved in vision transduction. Additionally, taste receptors share the use of the TRPM5 ion channel, as well as a phospholipase PLC-beta-2. Savory or glutamates. The TAS1R1 plus TAS1R3 heterodimer receptor functions as the savory receptor, responding to L-amino acid binding, especially L-glutamate. The umami taste is most frequently associated with the food additive monosodium glutamate, message, and can be enhanced through the binding of inosine monophosphate, IMP, and guanosine monophosphate, GMP, molecules. TAS1R1 plus 3 expressing cells are found mostly in the fungiform papillae at the tip and edges of the tongue and palate taste receptor cells in the roof of the mouth. These cells are shown to synapse upon the chorda tympani nerves to send their signals to the brain, although some activation of the glossopharyngeal nerve has been found. Sweet. The TAS1R2 plus TAS1R3 heterodimer receptor functions as the sweet receptor by binding to a wide variety of sugars and sugar substitutes. TAS1R2 plus 3 expressing cells are found in circumvallate papillae and foliate papillae near the back of the tongue and palate taste receptor cells in the roof of the mouth. These cells are shown to synapse upon the chorda tympani and glossopharyngeal nerves to send their signals to the brain. The TAS1R3 homodimer also functions as a sweet receptor in much the same way as TAS1R2 plus 3 but has decreased sensitivity to sweet substances. Natural sugars are more easily detected by the TAS1R3 receptor than sugar substitutes. This may help explain why sugar and artificial sweeteners have different tastes. Bitter. The TAS2R proteins function as bitter taste receptors. There are 43 human TAS2R genes, each of which, excluding the 5 pseudogenes, lacks introns and codes for a GPCR protein. These proteins, as opposed to TAS1R proteins, have short extracellular domains and are located in circumvallate papillae, palate, foliate papillae, and epiglottis taste buds, with reduced expression in fungi-form papillae. Though it is certain that multiple TAS2Rs are expressed in one taste receptor cell, it is still debated whether mammals can distinguish between the tastes of different bitter ligands. Some overlap must occur, however, as there are far more bitter compounds than there are TAS2R genes. Common bitter ligands include cyclohexamide, denatonium, prop, 6N-propyl-2-thiouracil, PTC, phenylthiocarbamide, and beta-glucopyranosids. Signal transduction of bitter stimuli is accomplished via the alpha subunit of Gust-Dusan. This G protein subunit activates a taste phosphodiesterase and decreases cyclic nucleotide levels. Further steps in the transduction pathway are still unknown. The beta-gamma subunit of Gust-Dusan also mediates taste by activating IP3, inositol triphosphate, and DAG, diglyceride. These second messengers may open gated ion channels or may cause release of internal calcium. Though all TAS2Rs are located in Gust-Dusan containing cells, knockout of Gust-Dusan does not completely abolish sensitivity to bitter compounds, suggesting a redundant mechanism for bitter tasting, unsurprising given that a bitter taste generally signals the presence of a toxin. One proposed mechanism for Gust-Dusan-independent bitter tasting is via ion channel interaction by specific bitter ligands, similar to the ion channel interaction which occurs in the tasting of sour and salty stimuli. One of the best researched TAS2R proteins is TAS2R38, which contributes to the tasting of both PROP and PTC. It is also the only taste receptor whose polymorphisms are shown to be responsible for differences in taste perception. Current studies are focused on determining other such taste phenotype determining polymorphisms. Sour. Historically it was thought that the sour taste was produced solely when free hydrogen ions, H+, directly depolarized taste receptors. However, specific receptors for sour taste with other methods of action are now being proposed. HCN1 and HCN4, HCN channels, were two such proposals. Both of these receptors are cyclic nucleotide-gated channels. The two ion channels suggested to contribute to sour taste are ACCN1 and TASC1. Salt. Various receptors have also been proposed for salty tastes, along with the possible taste detection of lipids, complex carbohydrates, and water. Evidence for these receptors is, however, shaky at best, and is often unconvincing in mammal studies. For example, the proposed INAC receptor for sodium detection can only be shown to contribute to sodium taste in Drosophila. Carbonation. An enzyme connected to the sour receptor transmits information about carbonated water. Fat. A possible taste receptor for fat, CD36, has been identified. CD36 has been localized to the circumvallate and foliate papillae, which are present in taste buds, and research has shown that the CD36 receptor binds long-chain fatty acids. Differences in the amount of CD36 expression in human subjects was associated with their ability to taste fats, creating a case for the receptor's relationship to fat tasting. Further research into the CD36 receptor could be useful in determining the existence of a true fat-tasting receptor. GPR120 and GPR40 have been implicated to respond to oral fat, and their absence leads to reduced fat preference and reduced neuronal response to orally administered fatty acids. TRPM5 has been shown to be involved in oral fat response and identified as a possible oral fat receptor, but recent evidence presents it as primarily a downstream actor. Types: Human bitter taste receptor genes are named TAS2R1 to TAS2R64, with many gaps due to non-existent genes, pseudogenes, or proposed genes that have not been annotated to the most recent human genome assembly. Many bitter taste receptor genes also have confusing synonym names with several different gene names referring to the same gene.
Please subscribe and thanks for watching.